and thank you all. Thank you all for attending. What happens usually is that I start sharing my screen and then you all disappear. So I can't see you. And if you want to interrupt, please do. And I'll be very happy to stop and try and explain something a little better. So I'll try and share now, and we will go from there. And I'll ask you at this point whether you can now see my screen. And if someone could let me know whether they can see the screen, that would be great.
Perfect, yes.
Okay, good. So lovely to talk to you today about the DecodeME project, which is a partnership between ourselves at the University of Edinburgh, the Charity Action for ME, and many people with lived experience of ME, funded by the MRC, the Medical Research Council, and the National Institute for Health and Care Research. So we're going to discuss today several things. Why have we focused on genetics? In terms of the project, where did it start? How did we get to the present situation? Then results, what they might mean, and what's going to happen next, and in particular for samples and data. And of course, very happy to answer questions during or after. So in terms of genetics, there are two main reasons why genetics might be important here. And the first is that it's now well known that pharmaceutical companies are needing genetic evidence for drug targets before they can proceed with the costly process of developing drugs. And they need that because genetic evidence doubles the likelihood of a successful progression through the development of a drug through to the clinic. So that's one point. The second point why we look at genetics in this particular way is that it's unbiased and it's comprehensive. Comprehensive meaning we look across the whole of the genome, and unbiased means that we don't have any preconceptions about what we will find. So we don't know what the biology is that we might find, and so any previous preconceptions are set to one side. And if you believe, as I do, that most aspects in biology we don't yet know, then it is a good way of proceeding. I don't think that we know everything yet in biology. And so starting with a blank canvas is a good approach. And then also what you know, which is that people with ME have symptoms that are across the whole of the body. And these fluctuate over time and in different people. And so it's not yet entirely clear where the root cause is of ME, in which system does it reside? And genetics can begin to address that point. So what did we know before DecodeME about genetics? Well, we knew some evidence that people from the same family can be diagnosed with ME. And then secondly, we knew that if someone in a family has ME, then the likelihood that someone else has ME doubles. And that is not just for close relatives, first-degree relatives, but also second and third-degree relatives, which gives it more chance, greater likelihood that genetics are involved. And to cut to the chase, when we measured this in DecodeME, we came up with a number, which I'm giving here as a percentage point of about 9.5% of something called heritability. And this is how much of the ME risk is predictable based on a variety of assumptions. It doesn't really matter what that number is, except for one thing. It isn't zero. So because we know it isn't zero, we know that genetics are involved. At the heart of DecodeME and any future project of whole genome sequencing, Sequence ME, at the heart of those projects are people with lived experience, people with ME and carers. And in all aspects of what we did, from management to the cohort delivery and the genetics, marketing and comms, all aspects, people with lived experience here in the darker color were part of the groups that were doing that. And I cannot emphasize enough how much scientific value that provided. I think that changed how we asked the questions, which improved the way in which people could respond to them. And it improved the numbers of people who wanted to volunteer to participate, because I believe, but tell me otherwise, with people with lived experience in the project, there was greater trust for participating in the project. And we needed to gain your trust, everyone's trust, to perform this project. And we needed, and as I'll show in a minute, not just hundreds, but thousands of people to be involved. So this is a complicated slide, and I apologize for that, but I've taken it from a genetics review, which is in the top right-hand corner. And it's got 3 different curves on it, all relating on the x-axis to time in the past, from 2007 to 2022. So I'm going to start with the blue, which is this first curve here. And these are the number of publications of this field of genetics that DecodeME falls into. And essentially, from 2007 onwards, there were hundreds of these publications afterwards, hundreds and hundreds. And all of these essentially were for other diseases and not ME. So that begs the question, why was it that ME was not used as a test case for this technology 10, 15 years ago? I think there are many reasons, and this perhaps would take another half hour to go over. But the key point now is that it is a technology that is mature. It should have been applied. And so we decided that it should now be applied, and its results should be accelerated towards the clinic. The green one is the number of signals that have been found in these publications. And around 5 or so, which is the green one here, at the beginning, and then that's jumping up to 10, 15, 20, up to 40. And that's because of the brown curve here, which is that the number of people involved in the study has gone up and up and up on average. So that's 100,000 people here. And with the larger number of people, you get more signals. And so if we draw a line here, which is at 18,000 samples, we have people's DNA from, we expect about 5 in the green here, 5 or so signals. So we'll come back to that number in a minute. So what is this type of genetics that we do? It's called GWAS, Genome-Wide Association Study. And it is at heart quite simple, in that at every place along our chromosomes where a person could have one or both, sorry, I'll say this again. A person could have one of these 2 letters, the C or a T. It asks the question whether people who with a particular diagnosis, here heart disease, are more likely to have C over T relative to everyone else, all of the other controls who don't have the disease. And so that at the heart of it is a statistical test saying, are there here at this one place, are there more people with ME or heart disease with a C relative to T, and when compared to controls? And because we do this at hundreds or in fact millions of places along the genome. We test each one of those. We need to have a very stringent statistical test threshold. And we also need a lot of people, as we said, and not just thousands, but in our case, over 15,500 for the initial results that I'll discuss. So 15,500 people with ME whose genetics we have read out, and we compare them against over 250,000 population controls that we didn't need to generate the data for because it was already available. So we spent our money on people with ME and not on the controls. So here's an example of what you could find in one of these genetic studies, one of these GWAS. And this is not for ME, it's for Lyme disease. And the results are set out on the x-axis on all of these different chromosomes labeled 1 to 22. And on the y-axis, it's essentially how significant the test is that I've just described. And there are 3 places where the significance rises above this horizontal line. This place on chromosome 4, which is labeled TLR1, place on chromosome 6, HLA, and a place on chromosome 11, where there's a lot of significance rising to this number 80 here. And the people, not ourselves, who did this study were very happy to see this because they saw 2 things that had been expected. They had expected this region of chromosome 6 to be involved because they had suspected that Lyme disease would be to do with infectious and autoimmune traits. And they suspected TLR1 because the TLR1 protein had previously found to be associated with this disease. But their discovery was for the 3rd place up here, for this place on chromosome 11, and it indicated this one gene with this complicated name. And this one gene, its product limits the growth of the bacterium that is involved in Lyme disease. So that immediately explained the susceptibility of people to Lyme disease at this place on chromosome 11. So how we got to where we are in DecodeME is that we, for about 10 years ago, we came together, people with lived experience and scientists. And our objectives were threefold. We want to identify the genetic differences in people with ME, because we felt that this would start to explain mechanisms. We wanted to build a cohort of people that other scientists could gain access to for future research. And we wanted to make sure that the data that we had collected, when people wish us to, we would give that data to academics and industry without people's identifying information associated with it. So we co-produced DecodeME with the community and recruited over 20,000 people over the age of 16. Everyone had to have a clinical diagnosis. They live in the UK, and their inclusion and exclusion criteria were consistent with 2 international sets of criteria, the Canadian and the IOM criteria. And we made sure that everyone had post-exertion malaise. So there were tens of thousands of people who were interested, completed the questionnaire, met the eligibility criteria, were given the saliva collection kits, and an amazingly high proportion of people who were given the kit returned their kit with the saliva collected in it. Not everyone's sample was able to have DNA extracted, and not everyone's DNA data turned out to pass quality control. And from this number, we have now alighted on 15,500 people that we initially are testing who have European ancestry. I'm just going to move where I am because where I'm staying in Oxfordshire, Other people have just arrived. So I'm going to move rooms to tell me if I'm losing signal. And underneath this, there are bar graphs which show that essentially the DecodeME people, the participants, are similar in ages and in comorbidities to what is expected from English people with an ME-like code G93.3. The kinds of symptoms that people in DecodeME have reported are very well known to you. Everyone had post-exertion malaise, but things like fatigue, muscle pain, noise susceptibility, confusion and brain fog, etc. All of those symptoms were highly prevalent among people in the cohort.
Yeah.
So I'm just pausing a little bit to say,
I'm watching the presentation.
these are the 8 genetic signals that we have highlighted in the preprint that was given out earlier on this month. So again, what we have here are the chromosomes from 1 to 22, and the higher these data points are, the more significant they are. And there is an industry standard to say that this horizontal dashed line, anything that's above that is significant at the genome-wide level. And you'll see that 6 out of 8 of them in this particular result are above the line. And these 2 chromosome 12 and 13 are slightly below the line, but they have risen above the line in other analyses. So we have added them. So there are 8 genetic signals. And what I've done here is to add a gene to label each one of these. Now, this does not mean that we know that this is the gene that confers the risk of people with ME. It is just a label. It is one of several genes usually that lies within the signal. And I'll come back to that about how we try and improve the candidature of genes later. So I said that in our primary study, we found 6 signals, and then there were 2 additional ones that were found in this other one, GWAS2, and one of those in something called infection. So GWAS2 we needed to do for technical reasons to allow us to do some replication in UK Biobank. I can answer questions on that, but it's for a technical reason. We did a study on females only, and we found 3 of these 8 signals in that. We found none for males only. And for those who said that they had an infection just prior to the first onset of their symptoms, we found 3 of the 8, and we found no signals among those who said that they did not have an infection. Now, we have to say that there may be good reasons why we found nothing among males and nothing among people without an infection just prior to onset. And that good reason would be that there are very few or relatively few people in those categories, providing less statistical ability to find these associations. So overall, what have we found in DecodeME? Essentially, the genes that lie in these signals point to 2 systems involvement. First of all, the immune system, and secondly, the nervous system. 2, and likely 3 signals relate to the body's response to infection. So, for example, BTN2A2, this signal on chromosome 6, there are a whole variety of very similar genes that together work to as a sensor of bacterial, of the bacterial infection. And then there's another one, OLF-M4 on chromosome 13 here, which the signal is next to that gene, and that its protein is involved in suppressing the immune system when there is an infection. So immediately with these signals, we could begin to find explanations for why people in particular have ME. There's another signal which is on chromosome 17, CA10, and that signal essentially, and I'll show this later, is what has already been seen for people not with ME, but people experiencing chronic pain. So what this is saying is that a previously found genetic signal is telling us back of what we have already heard for many years from people with ME about their symptoms of chronic pain. So that's the DNA speaking back to us of people's lived experience. So these are signals that align with how participants have described their illness. When we ask the question at these 8 different places, is there any statistical evidence for a bias towards females over males, for example? There was no such evidence. Now, we may yet find such evidence, and we may yet find such evidence in what we still need to do, which is to analyze the sex chromosomes, specifically the X chromosome. And we haven't yet done that. We had wanted to do it, but we ran out of time to do it. We have to consider it slightly differently from the other ones. And then we also asked our colleagues around the world to see whether these 8 signals had any evidence of statistical validation and replication in their data, in their ME-related data. And for half of the 8, there was indeed some degree of statistical replication, but I need to say it's quite modest. And I think there are good reasons for why it's modest, in that the ways in which people were brought together as a cohort in these different biobanks are very different from how we did this in DecodeME. So I'm going to say something about a technique called co-localization, which is quite complicated, but at heart is something that's quite simple, I think. And that is, we're wanting to ask the question whether our ME signals, and this is a signal here, this curve, that lies on top of the signal for another disease, such as this one. And on the left, they do not. They don't lie on top of each other. On the right, they do lie on top of each other. And so we want to separate those 2 possibilities and to have a statistical test that asks that question. And we use this twice. We use it first to ask whether the genetics of ME are similar to the genetics of other diseases, anything, to be honest. And then later I'll say, does the genetics of ME relate to the genetics of the activity of genes, which is a bit more complicated. So when we ask the question, is the genetics of ME, is that essentially the same as the genetics of other traits and diseases, we find that they are not. So on the 5 of these panels show our comparisons with things like chronic pain on chromosome 1 and depression chromosome 1, depression chromosome 13 and 20. And in those instances, although there are neighboring genetic signals for depression, they do not overlap statistically with the signal for ME. The one case where there is overlap is, and I've explained this already, on chromosome 17, there is significant overlap between the signal for ME and the previously found signal for chronic pain. So that's why I said that the genetics was telling us back what has been the lived experience of people with ME. We don't see all previously found pain genetic signals. So the one that was only reported 2 days ago in this paper on the top right, this is their genetic data I'm showing you. We do not see at all. It flatlines and there are no data points above this horizontal line. So for ME, we have no evidence that this genetic signal for pain intensity relating to this gene here, we see no evidence for that. And this is another benefit of the genetic study, in that although we obviously will prioritize biology for future investigation, we can also help to deprioritize investigations in the future. Now, this is an analysis using an algorithm called MAGMA. And what it does is essentially ask the question, is there a trend for where are the genes in the signal active? Where in the body are they active? So it takes all of the genes that it considers in the signals and asks, is there a preference for these genes being active in a particular place? And in DecodeME, the answer is very clear, that there are 13 tissues that are significant, and they're all brain tissues, 13 of them. So that provides the evidence for what I said previously, that there is evidence for ME being in part a neurological disease. So what might these results mean for people with ME and their family? So I need to say very clearly, I hope, that genetics is just one part of a larger puzzle. And the genetics that we have seen represent only part of what the genetics are that can, and in the future, I hope, will be discovered. So we're only seeing part of the genetics, and what we're not investigating is the environment. And by the environment, I mean things like people's infections that will be different because of different exposures. So that's the first thing. And the second thing I want to say is that all our results don't relate to any individual. They relate only to everyone in the cohort, everyone in the study. So they are results that relate to the average individual and not to any particular person. So what do the genetic results mean for someone's family if they have ME? They do not really mean anything in particular. They don't provide any evidence for a genetic diagnosis. And having all of the genetic signals that we have found could mean that you have ME, and it doesn't necessarily mean that you have ME. People with all 8 may not have ME. People without any of the 8 may have ME. So it's not, the results do not relate to any individual. The value doesn't come from helping individuals. It comes from pointing the future direction of where research should now be done. And we always said in DecodeME that there is a long journey that needs to be taken. And what we're doing here in genetics is trying to accelerate passage on that journey. In DecodeME, we are not testing whether people recover or not, whether their symptoms get worse or get slightly better. So we're not saying anything about that, and it doesn't say anything about treatment and testing. There might yet be some genetics of people with both ME and comorbidities like fibromyalgia, but we haven't yet done that. So in summary, the DecodeME results do not relate to individuals, and whether an individual has ME or not, it does not predict that accurately. And when we look at the signals, they cover several genes. So here's one example on chromosome 20. So you can see all these genes underneath, these little figures here. And so this signal could relate to any one of these genes. And so what we need to do is to prioritize them. So to prioritize them, we use this trick that I talked about before. We ask whether the genetics of ME overlap, they coincide with the genetics of how active a gene is. And here the activity is the amount of RNA that's produced, and that is in each individual tissue. And there are 50 or so of them. So we have to do this for each of the tissues. And then we get this, which I think is figure 4 in the preprint. So it's complicated. The tissues are on the bottom, the 50 or so, and the genes that lie in the signals are on the y-axis here. And there are large circles, and these large circles are where there is evidence for the coincidence of the gene signal and the ME signal. And where there is coincidence that is consistent with that gene being involved in ME risk. And the green ones are where the ME risk, when the risk is increased, corresponds to increased activity of the gene. The blue ones are where the increased risk is to do with decreased levels or decreased activity of the gene. So this helps us to say, for example, that if a particular gene is involved, do we need to make a drug that inhibits it or increases its activity? And that, therefore, is quite an important thing to know. So that's about gene prioritization. And we haven't completed all that we want to do on this, and we're doing this better. And so when we finish our project, we'll show the results, and they will be different from this. So what next? I've told you that we are going to do better gene prioritization. I've told you that we haven't yet done the X chromosome work. I didn't say anything about this part on chromosome 6, which is called the HLA and has been associated with immune function and autoimmunity. And that's why I haven't done that, is because we haven't done the best analysis that we could do using something called joint imputation. We do have some evidence that there might be a signal there, but we weren't confident enough in the result to place great emphasis on it in the preprint. We haven't yet done a replication test that we wish to do using ME cases defined out of the UK Biobank. We haven't yet done an analysis using everyone, not just people from Europe. So we'll use an algorithm called knockoff GWAS. And there are 2 other algorithms that we're already applying at the moment. We just don't have results from yet. Some of these computational runs take weeks to complete. And only when we have all of that will we complete the manuscript and submit it to a journal. And I hope that we will also have a revised preprint for everyone else to read. Now, we have in our freezers DNA samples, and that the residual DNA samples might be really helpful for future genetics. not to look at what we've been looking at, which are common genetic differences, but looking at rare genetic differences, because these rarer genetic differences are more likely to explain why ME runs in families, essentially. And that also might fast track our understanding of ME itself, which genes, specific genes, are involved. So that would be a separate project called Sequence ME. And why we haven't done that already is that it's really costly. This would cost somewhere similar to £18 million, whereas DecodeME costs £3.2 million. So we would use Oxford Nanopore technology sequencing because it will enable us to see parts of the genome that are completely invisible to the method that we previously used. We've done a pilot of 10 samples, and the first phase of this would cost about £8.5 million. So it's a very expensive project. Now, we also want to ensure that the value of the data lives on. So we always have encouraged researchers to apply for the data where the data is consented, and there is a process for this. And we've had at least a dozen or so applications, and we're working through those. Many of those applications have been processed and people have already received the data. So I hope it's clear that this is an enormous piece of work done by many people over many years. And these names are actually only a subset of all the people involved. And to all of these people, I give my thanks. So I'm very happy to take any questions, and I hope that was interesting to you.
Well, firstly, thank you very much for that, Chris. So what we'll do with questions is the first few we'll have that they will be recorded, and then we'll have a bit of time for any questions that people don't want for the face to appear on video. Should we go to Barry first and then Claudia?
Thank you, Chris. You were talking about things that are still to be done, including sort of very expensive, need very expensive funding. What are you funded for at the moment, and what are the prospects for finding the funding? The 18 million, I think you talked about.
Yeah.
And could you also give sort of best guesses as to timescales for the additional work?
Yeah. So the additional work would probably take another 3 or 4 years. It's a huge endeavor. It's even greater than DecodeME. DecodeME funding period is due to end this month. We will keep going. We have some funds that will keep us going for several months. So I pledge to you and to others that we will complete the DecodeME project, but it will be at a slower pace. Where are we looking for funds? Well, clearly these larger amounts mean that we need to go to government funding agencies and to high value philanthropists. So we're doing both of those things. To date, we have not raised a single penny, unfortunately. To start, we would need several million pounds. And so we're not going to start until we have that. The good thing to say about that, however, is that everyone is on board. So the people that we need to be involved, from charities to Oxford Nanopore Technology to our partner at the European Bioinformatics Institute, who would do the heavy lifting of the data analysis and managing, all of them are on board. and are very excited by this, if only it is to be funded.
Thanks for that, Chris. And Claudia.
Hiya. Yeah, firstly, thank you, of course, for all the amazing work. My first question was about the graph that was about whether the genes were like sort of up-regulated or down-regulated in the tissues. And I noticed the one to do with chronic pain was in like the prostate.
Prostate.
Only the prostate, which is obviously
Yes.
So
Yes.
could you explain that a bit?
So, good spot. Start off with. So clearly, what we're doing is data-driven, and we will do the same thing for all tissues and report all of the results. Do I believe that that prostate result is relevant to everyone? Well, actually, it's possible, and it is possible because there are commonalities of biology in different tissues. It's not that biology is specific to the prostate. There are other things that other places where the same biology exists. But I agree with you that the likelihood of that being relevant is lower than for the other associations. And that I don't also want to discount any gene that is not indicated in this analysis, because in a very complicated way, the data that we're using may not be relevant entirely. So the activity of the genes and their relevance may be greatest not at baseline, but when people have an infection, for example. And that's not that has not been used in the generation of the data that we have used for this. So in a very complicated way, even though this is the only way that we can prioritize genes, we do not think that this is a perfect way or a complete way.
Yeah. And secondly, if that's okay, just the slide you had about like sort of further work you'd want to do with DecodeME. Are you saying that you do have the funding to complete that work currently, just at a slower pace? Or is that sort of if you were to get further funding?
We have sufficient funding to take the work, to complete all of those things, take it to a version of the paper that will be submitted for publication.
Okay.
Now, I'm saying this to you because it has to be done. Where exactly we're going to get all the money required, I don't know, but most of it we have in hand, fortunately. And if we need more money to pay people to do the work, we will find it. It's important for us to do so.
Thank you.
And now to Fiona, please.
Hi, yeah, early on you mentioned chromosome 11 to do with Lyme disease. I just wondered, were you ruling out anyone that's had Lyme disease? Because I wasn't actually accepted into this. And I also noticed that chromosome wasn't discussed later on with your other data. I just wondered whether you were trying to keep the study simple. Oh, we don't know if this person's still suffering from Lyme or not, or whatever, keep them out of it in that way. But also there's obviously other infections, because I've had like issues with EBV and that, and you know, so yeah, it's kind of interesting that you mentioned it, but it's kind of not part of it. I don't know.
So we didn't exclude people based on Lyme disease. Let's be clear.
Okay.
That was just an example of what the genetics can do. But it's also true that the ME signals did not coincide with the Lyme disease signal.
Yeah. That surprises me, yeah.
And it did not coincide with the long COVID signal either. And I don't think that we can say immediately that that means they are different. And I think that because it's like an iceberg, we're seeing some things that are above the surface, but there's a hell of a lot more that we can't yet see that we would see if the study were to have been much bigger.
I mean, do you have any idea why some people weren't accepted on this then? I don't know, or what reasons might have counted people out, whether it was certain medications or certain comorbidities. I wasn't sure why I don't know.
Yeah, I can't say in individual cases, but the criteria that were applied are all in the preprint and its supplemental methods.
Right.
So again, I can't give reasons for individuals. And the value goes far beyond the genetics of DecodeME, though, just to say. And it was fantastic that everyone who did participate and complete the questionnaire, that you and others did so, because we gained hundreds of thousands of years of lived experience from that we can give all of that data where there is consent to others to pore over.
Oh, it's definitely valuable data. It's very interesting. Thank you.
If we can go to Julie next, please.
Hiya. Obviously, thank you, Chris and the team. I think this might be to do with the iceberg you've just mentioned. So I understand how you can have all 8 genes and not have ME. I don't understand how you can have ME but not have any of the genes. Is this because you haven't looked at everything under the surface of the iceberg, or?
Yeah, there's so much that I would still love to know. There will be many more signals found when DecodeME is done with a larger number of people. That's what has always been seen for other diseases. Other diseases have gone to hundreds of thousands of people and have begun to find not 8, but 80 type signals, giving far greater resolution of the biology. That's what's needed. And then the technology that we used was only looking at these common variants, that is DNA letters that are commonly different among us in our population, but not the rare ones. And so there will be a lot, I think, to be found in the rare variation. So I will not rule out I cannot rule out based on genetics whether or not someone has ME. It's not just the genetics; it's the environment that interacts with the genetics in very complex ways.
Yeah, I was trying to get my head around it, and I thought, well, just when you then went on and it said something to who was speaking last, it started to make a bit more sense. I'll ask another question. So, obviously, Mel was a little bit involved with the delivery plan and the task and finish groups and so on, and obviously witnessed the very disappointing result for the research section of that. And I'm just wondering if you think that if DecodeME had been prior to the publication of the delivery plan, if the research situation would have looked different, and also if that would perhaps be easier to revisit that now, because we're meant to be still looking at the delivery plan going forwards. So do you think, is there a possibility that's going to have an impact on funding coming from MIR and so on and so forth?
Yeah. So the funding source would be the Medical Research Council, because the NIHR essentially fund studies to do with translation to the clinic. So we're still in the discovery phase. So the discovery phase funder is the MRC. We invited the MRC and the NIHR to all of our scientific advisory board meetings throughout the whole of the study. They knew what was going on all the way through. MRC, and I meet with them fairly regularly now, essentially say, you've got to compete with all other diseases. And I tell them it's impossible, because the evidence base doesn't exist. And so things that we know much more about, cancer, provide a more understandable, cogent case for funding. And of course, there is a preference for it then to be funded. There is not a level playing field. I have not won that argument. I've also said to the MRC that they need to look at themselves very carefully as to whether or not, and I believe it is the case, that they are funding female biased disease less than they are male biased diseases. They need to answer that question, and they seem to be dodging that question quite well at the moment. The amount of funding that they distribute has gone down, and so they are basically saying that they don't have enough money to, and they say that they don't prioritize diseases for funding, and that's not true. That's what they say, but they clearly will not prioritize ME. And my question to them always is, why not? Not just that for them to tell me that they will not, but my question is, why not?
And do you ever get an answer?
No, but I think it's the right question.
Yeah, it is the right question. Yeah, definitely. Thank you for asking that question. It's very frustrating for us all, obviously. Thank you very much, Chris.
Thank you, Julie.
If we could go to Caroline next, and then Barbara, and then what I think we'll do is stop the recording and give a chance for anyone who wants to ask a question off camera.
Thank you. Thank you. I hope I'm not the only one whose head is slightly spinning at all the science. So how do we, as non-scientists, talk about this? It's not that there are 8 genes. It's because you use your language very carefully because you know what you're talking about. So you talk about 8 signals, and is that 8 signals in each of 8 separate genes? But it's not the whole gene by any means, is it?
Yeah. Okay, so I'm going to, at risk of your head spinning some more, tell you that the genetic differences often get co-inherited down generations. So they stick together down generations. And that means that we can't really tell whether it's this one or this one because they go down the generation together. And that's a problem. And it's a problem that is difficult to solve, but we are addressing it. But it means that if there's a genetic difference in this gene, genetic difference in this one, and they always go together in people, we can't tell them apart. So that's why I use the word signal, which is the overarching signal that includes both of them. And I don't say this is a single gene. And in at least one of the signals, there is evidence that contributing to that signal are 2 subsignals. So it's there are, and they coincide, which means that we should believe them more. So sorry to say it's complicated.
Okay, thanks very much.
And if we could go to Barbara, please. You've been waiting patiently with your hand up.
Thank you. I'm going to roll back quite a few years, Chris, and thank you for your amazing work, to the issue of allergy being ectopic, and the research that was done that suggested 60% of people that receive a diagnosis also have a history of allergy. And going back to your colleague, Stephen Holgate from Southampton, who is involved with the Medical Research Council, he produced a really important paper with someone from Cambridge, I forget her name now, called Allergy, the Unmet Need. So my question is that as ME has historically and evidenced in the research had a huge overlap and comorbidity with allergy, and bearing in mind allergy has always been a correlation, even though Stephen Holgate has worked in this for many years. Stephen Holgate also did the research with the late Jonathan Kerr on the gene expression work as well, that implicated environmental factors like pesticides. And Stephen Holgate was a member of the Royal Commission on Environmental Pollution that looked at ME in 2005 with their report on people like me who live in rural areas that have this added burden of environmental impact. So my question is there any merit in going back to looking at this overlap with allergy, getting Stephen Holgate on board from that angle, wrestling the funding committees at the MRC away from the one where you know who, I don't know if he's still involved in triaging the research, Mr. Wesley. I know he was on one of the funding panels going back a long time. And perhaps applying for funding from a different work stream within the Medical Research Council.
All of that was excellent. Thank you so much. So the genetic signals for allergies are often in this part of chromosome 6 that I've called the HLA. So from our point of view, it is incredibly important for us to do that analysis to the best of our ability. We did see a signal, but we're not as confident in it as for the others. When we first saw that signal, we immediately went to the immunologists and told them about it. And we started cooking up a plan to get funding to follow up on that signal. But ultimately, we together decided that we should wait until the best possible analysis has been done before proceeding. In other words, to have the greatest evidence and therefore the most solid research plan that has the greatest likelihood of being funded by the MRC. Stephen Holgate has retired. He's still working in part, but he's retired. But there are many immunologists that I've come across, and I will always push them to get involved. And there are other funders I know, like Versus Arthritis, who would, I think, be able to pick up on this.
Fiona, if you could just stop the recording then. So we're getting very near the hour.